Assalamualaikum. Hi. So we continue with the Bioreactor Engineering Part 1, where in this presentation, it will be on the stoichiometric parameters in the growth kinetic and the stoichiometric calculation based on elemental balance. So you can refer to the textbook written by Schuller and Cargi, the Bioprocess Engineering Basic Concept, second edition by Prentice Hall. You can read in chapter 6 and 7. Let's start with the stoichiometric parameters in growth kinetic. For this subtopic, you can refer to the textbook at chapter 165. Okay, then let's start with the yield coefficient, where the yield coefficient is defined based on the amount of consumption of a given material. So for example, like here, it's on the growth yield, denoted by Yx over S, the yield coefficient of cell concentration over substrate, which is, you can calculate, okay, by using this formula, which is equals to minus delta X over delta S. Delta here means the difference between the initial concentration with the final concentration. So, in order for you to calculate the growth yield or the yield coefficient of cell concentration over substrate, okay, you need the value of initial concentration of cell and the final concentration of cell after the fermentation, and the initial concentration of substrate, okay, and the final value of substrate after the fermentation. Alright, so minus signs here means because of the growing of the cell, okay, the cell concentration is keep on increasing, okay, as time, but the substrate is decreasing. Alright, that's why there is a minus sign in front of the equation here. Okay, at the end of the batch growth period, okay, we have an apparent growth yield or observed growth yield. Okay, substrate may be consumed as following equation. Alright, so remember that the substrate that being added in the fermentation, it will keep on reducing as the cell is growing and the product is forming. Okay, alright, yield coefficient is based on other substrates or product formation, which can be defined by, if it's over oxygen, it will be Yx over O2 equals to minus delta X over delta O2. Okay, here also we have this minus sign because of the uptake of the oxygen based on the initial value of oxygen being introduced in the fermentation. Okay, if you have to calculate the yield coefficient of product over substrate, then you can use the equation here, Yps equals to minus delta P over delta S, because of the delta S is, okay, reducing, the substrate is reducing, right, as the product is forming, the formation of product, and the cell is increasing. That's why this is there is a minus sign in front of the equation. Okay, for organism growing aerobically on glucose, which is Yx over S, okay, is typically 0.4 to 0.6 gram of cell per gram of substrate for most of yeast and bacteria. Okay, while the yield coefficient of cell concentration over oxygen consumption is 0.9 to 1.4 gram of cell over gram of oxygen consumed. Alright, this is as a reference. Okay, if we want to know the details, we need to calculate based on the data given. Okay, so anaerobic growth is less efficient and the yield coefficient will be smaller. You can see in figure 6.5 in the textbook. Now, can we consider oxygen as the substrate for the bacterial growth? The answer is yes, because it is for aerobic growth, then the oxygen is required. This is figure that shows the evaluation of observed yield in batch culture for cell, substrate and product concentration. So if you have a graph of X over S, you may able to find the yield coefficient for X, Y, XS. Okay, if you have the graph of P over X, then you can find the yield coefficient of YPS. Okay, then same goes to YPS here if you have that P and S value. Okay, so and this is the observed the apparent yields in batch culture for cell, substrate and product concentration. Okay, as you can see from graph A, it has a negative slope. Okay, where the substrate is consumed and become less. Okay, the cell concentration X is increasing. And from graph B, product formation is directly proportional with cell concentration and the slope is positive. For graph C, it is almost to graph A, where the substrate ceases decreasing as the rate of production increases. Stoichiometry of microbial growth and product formation. So as the cell growth and product formation are very complex process, it reflects the overall kinetics and stoichiometry of the thousands of intracellular reactions that can be observed within the cell. Although it is complex, the stoichiometry of conversion of substrate into products and cellular materials can often be represented in a simple pseudo-chemical equation. Okay, let's take a look on the definitions. Let's recall on YMXS, which is the overall growth yield coefficient when no maintenance is considered. By comparison, the ATP yield coefficient, which is denoted by YX over ATP, okay, represents the amount of biomass synthesized per mole of ATP generated. Okay. A maintenance coefficient can also be estimated by using the equation analogous to that developed for substrate yield coefficient in the chemostat. Can refer to equation 113 here, okay, where Y apparent X over ATP is the apparent yield of biomass per mole of ATP generated, and MATP is the rate of ATP consumption for maintenance energy. Okay, table 7.1 shows the growth parameters of some organisms growing anaerobically in the chemostat. Okay, similarly, yields based on oxygen consumption. It can be defined and calculated by using equation 114. Okay, you can see here MO2. Alright, so the maintenance energy, it can be based on the oxygen or the substrate or the energy required in order for the cell to survive and to repair themselves for survival. Information from some measurements can be usefully combined to derive important parameters. For example, the respiratory quotient, which is denoted by RQ, which is defined as the moles of CO2 produced per mole of oxygen consumed. Okay, moles of CO2 produced per mole of oxygen consumed, RQ. Then there is the ratio of phosphate bonds formed per unit of oxygen consumed, which is called as PO ratio. Okay, phosphate over oxygen. A closely related parameter is proton over oxygen ratio. It gives the number of H plus ions released per unit of oxygen consumed. Electron generation is directly related to proton release. Usually 4 mole of electrons are consumed per mole of oxygen consumed. The generation of electrons results in the expulsion of X and H plus that can be used directly to drive the transport of some substrates or to generate ATP, meaning that whenever the cell is consuming the oxygen for growth, the cell might as well producing CO2 like we human being, which also generating CO2. Okay, the complex of mass and energy balances for cellular growth can be decreased greatly through the recognition that some parameters are nearly the same irrespective of the species or the substrate involved. Okay, these parameters are referred to as regularities. Okay, three important regularities need to be considered here. Okay, you can see here the listed regularities. Okay, and further, yx over e, okay, is equals to the number figure here, which equivalent of electrons, and these observed average values facilitates estimate of other growth related parameter.